Have you ever wondered what actually controls cell division? Deep inside every cell in your body, right now, as you watch this, there are tiny structures, working silently, making sure your cells divide correctly. And if they fail, the consequences can be serious. Today, on Gray's Gyan, we are going to explore three of the most fascinating, and often misunderstood, structures in cell biology. Centrioles, centrosomes and basal bodies. These are not just vocabulary words for your exam. These are real, living molecular machines inside your cells. And by the end of this video, you will understand exactly what they are, what they do, and why they matter. So let's dive in. Before we go into the details, let's get the big picture. There are three main structures we are going to cover today. First, centrioles. Think of these as tiny hollow cylinders made of protein tubes. They form the core of the next structure. Second, centrosome. This is the cell's main microtubule organizing center. It is the command center that controls where microtubules go, and it is responsible for pulling chromosomes apart during cell division. Third, basal bodies. These are very closely related to centrioles. In fact, they are believed to be derived from them. Their job is to anchor cilia and flagella, those hair-like structures you see on the surface of certain cells. These three structures are deeply connected. Understanding one helps you understand the others. So let's start with the most fundamental, the centriole. Imagine a tiny barrel. A hollow cylinder, so small that you cannot see it even with most microscopes. That is a centriole. To give you a sense of just how small we are talking, a centriole is only 0.2 micrometers in diameter and 0.4 micrometers in length. For comparison, a single human hair is about 80 micrometers wide. So a centriole is incredibly, almost unimaginably small. Now, what is it made of? The wall of this barrel is formed by 9 groups of microtubule triplets, arranged in a ring. Each group has 3 microtubules bundled together, and these 9 groups are connected to each other by several linking proteins. Picture a barrel with 9 staves, where each stave is made of 3 protein tubes. That is your centriole. Now, here is something important. In almost all animal cells capable of dividing by mitosis, you will always find at least 2 centrioles. Never just one. They are found in pairs. These 2 centrioles lie at right angles, or most commonly at an oblique angle, to each other. This paired arrangement has a special name. It is called a diplosome. One exception worth noting, eggs. Eggs undergo meiosis, not mitosis, and they actually lack centrioles. Exam tip, remember, 9 triplets, always in pairs, arranged as a diplosome. These are common MCQ points. Now that you understand centrioles, let's zoom out and look at the centrosome. The centrosome is a densely filamentous region of cytoplasm, a kind of organized, concentrated zone, found right at the center of the cell. It houses the centrioles, but it is much more than just a container for them. The centrosome has 3 major functions. Function number 1, it is the major microtubule organizing center, or MTOC, of most cells. This means the centrosome is the factory where new microtubules are produced. Microtubules are long protein filaments that act like roads inside the cell, helping transport materials and giving the cell its shape. Function number 2, during cell division, the centrosome generates the mitotic spindle. You can think of the mitotic spindle as the machinery that grabs chromosomes and pulls them apart to opposite ends of the cell. Without the centrosome doing its job correctly, chromosomes could be distributed unevenly, a serious problem. Function number 3, the centrosome influences the position of the Golgi apparatus. Because the centrosome sits at the center of a network of microtubules, and because certain membrane vesicles use motor proteins to travel along these microtubules, the Golgi apparatus naturally concentrates near the centrosome. This positioning is actually important. It helps the Golgi send its vesicle products to the right parts of the cell efficiently. Think of the centrosome as both a factory and a traffic control center inside the cell. Let us talk about something that is absolutely crucial to understanding how the centrosome works, and that is microtubule polarity. Microtubules are not symmetrical. They have two distinct ends. One end is called the minus end, and the other is called the plus end. Here is the key point. In a cell, all microtubules that radiate from the centrosome have their minus ends pointing inward, toward the centrosome, and their plus ends point outward, away from the centrosome. Think of it like the sun and its rays. The centrosome is the sun. The microtubules are the rays of light spreading outward. All rays begin from the sun, that is the minus end. And they extend outward into the cell, that is the plus end. Why does this polarity matter? Because motor proteins, like dynein, use this polarity to know which direction to travel. Dynein moves toward the minus end, which means it moves things toward the centrosome. This is how membrane vesicles, including those from the Golgi apparatus, end up near the centrosome. This elegant system allows the cell to precisely control where materials are delivered, to specific regions of the cell, at the right time. Now let's talk about how centrioles multiply. This process is called centriole biogenesis, and it is remarkably precise. It happens in 4 steps, timed to the cell's cycle. Step 1, S phase begins. This is the DNA replication phase of the cell cycle. The moment DNA starts to be copied, a signal also triggers the duplication of centrioles. Timing is everything. Step 2, daughter centriole forms. A new, smaller daughter centriole begins to grow at a right angle to each existing, maternal centriole. It grows out perpendicularly, like a branch from a tree trunk. Step 3, mother-daughter pair. Each mother centriole paired with its new daughter centriole forms one pole of the mitotic spindle. So when the cell is about to divide, each half of the spindle is anchored by a mother-daughter centriole pair. Step 4, full maturity. The daughter centriole does not fully mature immediately. It reaches complete maturity only just as the progeny cells, the newly divided daughter cells, are about to enter the next mitosis. This carefully timed duplication ensures that every daughter cell receives exactly 2 centrioles. Not more, not less. Key concept: centriole duplication begins at S phase, the same time as DNA replication. This synchronization is vital for accurate cell division. Gametubulin, the nucleation specialist. Let us now talk about a very specific and important protein inside the centrosome, gametubulin. The microtubule organizing center inside the centrosome contains complexes of gametubulin. Gametubulin is not the same as the alpha and beta tubulin that makes up the body of microtubules. It has a very specific job, nucleation. So, what does nucleation mean? Think of it like lighting a match. Before a fire starts, you need a spark. Gametubulin is that spark. It provides the initial trigger, the template, that starts the assembly of a new microtubule at its minus end. Without gametubulin, the tubulin proteins would just float around the cell with no way to organize themselves into a proper filament. These gametubulin complexes are embedded in the pericentriolar material, abbreviated as PCM, which is the dense, cloudy region of protein that surrounds the centrioles within the centrosome. So to summarize, the centrioles are the structural core. The pericentriolar material surrounding them contains gametubulin, and gametubulin nucleates new microtubules at their minus ends. All of this together makes the centrosome the powerful microtubule organizing center that it is. Remember, alpha and beta tubulin build the microtubule body. Gametubulin starts the process at the minus end. Basal bodies, anchors of cilia and flagella. Now let's meet the third structure, basal bodies. Basal bodies are microtubule organizing centers that are very closely related to centrioles. In fact, it is believed that basal bodies are actually derived from centrioles. They share the same fundamental structure, a ring of 9 microtubule triplets. But basal bodies have a specific location and a specific job. They are found at the base of cilia and flagella, and their job is to anchor these hair-like projections to the cell surface. Now, let's understand the structural connection between basal bodies and cilia. The axoneme, which is the internal skeleton of cilia and flagella, has the classic 9+2 arrangement. 9 outer microtubule doublets surrounding 2 central microtubules. Where do those outer doublets come from? They originate directly from the basal body. Specifically, 2 of the 3 microtubules in each triplet of the basal body continue upward to become the doublets of the axoneme. The third microtubule in the triplet, called the C-tubule, does not continue into the axoneme. It stays at the base. So the basal body is not just an anchor, it is the foundation from which the cilium or flagellum literally grows. Structural summary: basal body equals 9 triplets. Axoneme equals 9 doublets plus 2 central. The outer doublets of the axoneme grow from 2 microtubules of each triplet in the basal body. Let's now bring all 3 structures together and compare them side by side. Looking at structure, both centrioles and basal bodies are made of 9 microtubule triplets. The centrosome is larger. It is made of centrioles plus the surrounding paracentriolar material. Looking at function, centrioles form the structural core of the centrosome. The centrosome organizes microtubules and assembles the mitotic spindle. Basal bodies anchor cilia and flagella. Looking at location, centrioles live inside the centrosome, at the center of the cell. Basal bodies are found at the base of cilia and flagella on the cell surface. Looking at which cells contain them, centrioles and centrosomes are found in all dividing animal cells. Basal bodies are found specifically in cells that have cilia or flagella. And finally, basal bodies are believed to be derived from centrioles, making them structural cousins. Take a moment to study this table. It is a great revision tool, and these comparisons are a very popular source of exam questions. Alright, let's quickly revise everything we have covered today. Point 1. Centrioles are hollow cylindrical structures made of 9 microtubule triplets. They are always found in pairs, the diplosome, in all dividing animal cells. Point 2. The centrosome is the centrioles plus the surrounding paracentriolar material. It is the major microtubule organizing center of the cell. Point 3. Gametubulin in the centrosome nucleates new microtubules at their minus ends. It is the spark that starts microtubule assembly. Point 4. The Golgi apparatus concentrates near the centrosome because motor proteins like dynein travel along microtubules toward the minus end, toward the centrosome. Point 5. Basal bodies are derived from centrioles. They anchor cilia and flagella to the cell surface, and the outer doublets of the axoneme originate directly from the basal body triplets. Point 6. Centriole duplication begins at S phase, in synchrony with DNA replication. The daughter centriole matures fully just before the next mitosis. And that is a wrap on centrioles, centrosomes, and basal bodies. I hope this video has made these concepts clear and easy to remember. Gray's anatomy can feel overwhelming at times, but when you break it down, step by step, it all makes sense. If you found this helpful, please like this video and subscribe to Gray's Gyan. Your support means the world, and it helps this channel reach more students like you. Drop any questions you have in the comments below. I read every single one.